Good morning, ladies and gentlemen. It's Mr. Citron. Clearly, I'm not here. I'm at jury duty today, so just thought I'd record a short video for you and let you know what it is you need to accomplish. And hopefully, the substitute will help us out. And thank you and good morning. And so here's what we've got planned. We have finished mitosis. Any of you guys who need to submit chapter 10, page 300, please do so to the sub, and they'll take care of that and make sure I get it when I return. We have wrapped up mitosis, where we have actually created two identical daughter cells going through the interphase, obviously, prophase, metaphase, anaphase, telophase, and then cytokinesis. We are skipping ahead now to chapter 11, section 4, and in that particular section, we're talking about meiosis, meiosis is when we are actually creating not identical daughter cells, but unique daughter cells. And this happens in a very isolated event. It's during sexual reproduction. So during those, and we're creating those gametes, those offspring that we want to create, so that the offspring is going to be genetically unique. As I start to incorporate new things, of course, we come with new vocabulary. The vocabulary is coming up on a slide, and in that slide I'm going to ask if the substitute could pause the video at that point. You guys will take about 10 minutes, 7 to 10 minutes or so to get those words copied down, and then because you have your books, wink wink hint hint, you'll have that time, you'll be able to get those terms defined. My books are locked up. The substitute does not have access to those books. I didn't leave my keys. So you guys are on your own for books. If you don't have them, make sure you get the words jotted down, and you're going to need to complete that on your own time. Pause the video when those images come up, and then as soon as you're done, take about 5 to 10 minutes, we'll restart the video and we'll be on to step 2. Alright, well, you've had some time to do that vocabulary, and now what we've got is a transition. So we're actually going to look at meiosis, and I'm going to ask you very quickly to look at this document, and again, thank you, substitute, for if you feel comfortable in running this discussion, by all means. But ladies and gentlemen, take a look at this picture that's going to flash up on the screen, and as you're looking at it, think to see if you can figure out who produced which children. And I'll tell you, the top line are the adults, and they were from probably the 60s or so, and then the second line is their children, which are from maybe the 80s, early 90s or so. And the two on the top row, or the four on the top row, they each got married and then produced 2 children each. And let's see if you can look at it to see if you can follow where the different images and where the different traits carried through from the parent generation down to the offspring. So take a look at this diagram. And so as you're taking a look at this picture, you want to look to see how offspring 1 and offspring 2 and offspring 3 and offspring 4 compare to, or are similar to each other, and then how they compare to the adults. So take a careful look, looking at facial structures, looking at hairlines, looking at smiles, looking at bones and the breadth of nose and all of those other. Alright, by now you probably have some opinions as to who offspring were produced from whom, and you can see that in couple 1, they're both, well, in both couples there is an Asian male and then there is a Caucasian blonde female, and in all of the offspring you get a blending of all those traits. And they have child 1, female, with kind of an, looks like maybe a blondish hair with darker tones. Female 2, or organism 2 is female, and then there's a young boy, and then there's another young boy. So 2 boys, 2 girls. And I will tell you, because I know the answer, and you come up with your own conversations about the way in which traits get blended, that child 1 and child 4 are brother and sister, and child 2 and 3 are brother and sister, and children 1 and 4 are from couple 2, and 2 and 3 are from couple 1. So if you got that right, congratulations. If you didn't, it's no big deal. But we're going to be looking at the way in which those offspring, and especially the gametes that they produced, are actually going to be creating to create unique individuals. So as you look at the next diagram, we've transitioned from the actual children and the parents and taking a look at some more of the hardcore biology behind it. I keep using this term gametes. Gametes are the types of cells that are created when you go through meiosis, and they are the types of cells that you are creating through to create half your normal number, so that when they recombine with the parent, the other parent, then you get a full complement of chromosomes. Humans have 46 chromosomes. That is your normal number, sometimes referred to as your diploid number, 46. When you are about to, or when the organisms are going to contribute a gamete cell, which could be a sperm or an egg, they are going to have half the normal number of chromosomes. It's going to be haploid. Haploid of 46, or half of 46, would be 23. So if we take a look at this circular diagram that we have here, we've got ovaries and testes, which are producing the gametes for males and females. Those cells go through meiosis. Then you create an ovum and a sperm. Then at fertilization, there becomes a zygote, which is what is a combined egg and sperm. That number then goes, the chromosome number now again goes from 23 from mom, 23 from dad, you get half and half, to 46. So that's your overview, and then of course the organism does go through all the mitotic divisions that we've talked about. Every cell goes through mitosis. Your skin cells, your nerve cells, all those cells to grow as the organism gets larger to an adult form, and that's why that circle continues around until a point where you're going to actually create multicellular diploid adults. You could, and the word is proper and correct, to refer to your adult parent as diploid. Just do it carefully. Don't walk into mom and dad and say, "Hey, diploid." But if you're referring to genetically, they are all of their cells. All of your cells, as a matter of fact, are diploid. You are a diploid. I'll pause for the laughter that surely will ensue. Diploid. It's fun to say. It does describe, though, the normal number of chromosomes that you have in all of your cells. 46. That is the normal number of chromosomes that you have. You are diploid. So from now on, I think I might come in and say, "Good morning, diploids." I mean biologists. I mean students. And you'll be able to get a sense of what's going on, at least regard to your genetic number. So we're going through some of the steps of meiosis, and we kind of enlarge this diagram. We're going to see that meiosis has some steps. Guess what? They're interphase, prophase, metaphase, anaphase, telophase. And we're going to go through that process twice, and we're going to follow, again, what happens to the DNA. We're not going to map what happens to the nucleus and the cytoplasm this time. It's not as essential because you already know that. You know what's going on with the nucleus. You know what's going on in the cytoplasm from mitosis. We're just going to double it up in meiosis. All right, we're kind of in the meat of it now, where we've actually got meiosis 1 and meiosis 2 taking place. There are actually two phases within there, and each subphase has, of course, its own phases. So meiosis 1 has an interphase, a prophase, a metaphase, and an anaphase. And in those phases, it's all exactly the same as mitosis. There's no difference there. We have the centromeres, the centrosomes, the spindle fibers, we line them up, we have sister chromatids, and the only difference here is that we actually have homologous chromosomes now. And at this point, during these prophases, these homologous chromosomes will pair up and kind of exchange little segments of DNA. So they're not exactly identical segments of DNA that get passed on to the offspring. This is important because this is what makes you uniquely you. So if you look at the prophase 1 image, you'll see that you've got your spindle fibers are forming, you've got a tetrad, which is just the 4 chromosomes lined up next to each other. We've got our sister chromatids still there, just like we talked about in mitosis. But we get little segments that kind of get shunted from one side to the other. This is a process known as crossing over. And when this happens, small segments from one piece of DNA get carried over to the other piece of DNA. And so that's why you don't have 100% identical offspring, because everything's just slightly different. The rest of it's the same. We get microtubules attaching, the spindle fibers we've called them. We have a metaphase plate or the equator. That's where they line up in metaphase. In anaphase, they start to pull apart. At this time, though, rather than pulling apart each of the sister chromatids in anaphase 1, we're pulling apart each of the homologous chromosomes. So if you look carefully at that anaphase 1 picture, all the blues and reds that are drawn there, they're still pretty much intact. And as we transition to the next image, you'll see that meiosis 2, we actually go through a telekinetic telophase and a cytokinetic phasing of splitting them, but then we end up going through the entire process once again. And that is where we go to metaphase 2, anaphase 2, and now our sister chromatids can be pulled apart. Each of those remaining cells then has half the normal number of chromosomes. They are haploid. Remember that is that haploid starts with an H, half starts with an H, half the normal number of chromosomes, haploid. And all of those offspring, all those resulting cells, excuse me, not offspring, they're gametes, all those resulting cells come from meiosis, are half the normal number of chromosomes. There are 4 of them this time, because we've gone through this process twice, and they have unique segments of DNA. And once again, you can see the reds and the blues are actually interspersed rather than being all red and all blue. So as we move to the next image, we've got possibility 1 and possibility 2. And each are 2 equally probable arrangements of chromosomes during metaphase 1. You can line everything up across that metaphase plate right in the middle of the cell. Remember, metaphase means middle. And those genes are going to line up, all those chromosomes and their tetrads and their homologous chromosomes paired up. Going to line up right in the middle. Skipping down to metaphase 2, which is the next bubbles there, those next cells, you'll see that we can split off all the blue or all the red, or we can have a mixed match of the blue and the red. Again, either way is equally probable. It doesn't necessarily have to be one way or the other. If we look at the gametes, so these are the sperm and the egg down here at the bottom, the combination 1, the combination 2, in combo 1 you've got all blue. Combo 2 you've got all red. Combo 3, you've got a little blue, little red. And in combo 4, you got a little red, little blue. Of these, and this is all possible. So if any of these arrangements possibly happen, what you end up with are different possible offspring from the same parent. So you could end up with these gametes. All of these could be dad's sperm. These are all the same. These all could be dad's sperm. That is why you and your siblings don't look identical. You are not. And even though you might have some of the same genes and you might look a lot like mom, a lot like dad, you can't be identical, because as their gametes are formed, as they go through meiosis, those little segments of chromosomes get crossed over during that prophase when they're all getting moved around. And by getting them crossed over, you end up with variations in the genes of your parents. So when fertilization occurs and you are created, you get some from mom, some from dad. Dads are mixed up even amongst dad's own sperm. They're not all the same. Moms are mixed up even amongst all her own eggs. They're not the same. And so that recombination can show you why you might look very much like your brother or sister, or you may look almost just the faintest little bit of, is that my true biological sibling? And now if you have a stepsister who shares a mom or shares a dad or something of that nature, well, clearly you've got a different parent contributing to the mix. You might have a similar trait that with the shared parent, but it's not going to be 100% concrete, and you're never going to look identical to a half sibling. Very similar, perhaps, but never identical. Alright, and finally, to wrap up for today at least, you'll see this image next is a mitosis and meiosis comparison. So we actually get 2 different segments here, a side-by-side comparison of how we go through prophase, metaphase, anaphase during mitosis, which you know about, and how we go through meiosis, meiosis 1, meiosis 2, and we show metaphase, anaphase, and then the whole set of meiosis 2. And mostly what this image is showing you is it's showing you the difference in the formation of the daughter cells. In gametes or meiosis, you create 4 haploid daughter cells, and in mitosis, you create 2 diploid daughter cells, and those diploids have the normal number of chromosomes, like you, and haploids have half the normal number of chromosomes, and that's why we have the formation of those gametes. That is your significant difference. So I'm going to ask that we stop the video here, and as we're on this slide, we can pause it for a moment so that you can get just a jotting of some of the key things from this particular image, and then we can move on to the next tasks for the day. Thank you very much for your attention. I hope that you are acting, I'm sure that you are acting politely for our substitute, and I look forward to a good report, and hopefully I will see you tomorrow, and if not, then we'll have a second segment via camera if we can get it accomplished. We'll see how much time I have after being in the jury all day. Thank you, ladies.